Hi, this is Alex from devstories.eu. Scanpy Single Cell Analysis, Episode 5 of 16. Out of the roughly 30,000 genes in the human genome, most are just doing basic cellular upkeep. If you try to analyze all of them at once, the sheer volume of baseline activity will drown out the actual biology you're looking for. To find the real signal, you need feature selection and highly variable genes. First, a quick distinction. You have already used a basic filtering function to drop genes that are barely detected across your dataset. That step cleans up technical noise and empty droplets. Highly variable gene selection does something entirely different. It assumes the remaining genes are real, but asks which of them are actually informative. Think of it like this: sifting through tens of thousands of genes to find the ones that drive differences between cell types means discarding the boring baseline housekeeping genes. A housekeeping gene is active in almost every cell at roughly the same level. Its expression is stable, which makes it useless for distinguishing a T cell from a B cell. We want genes that are heavily expressed in some cells and totally silent in others. These are the highly variable genes. Usually, you want to narrow your dataset down to about 2,000 of these key drivers. In Scanpy, you handle this with the highly variable genes function, but you cannot simply rank genes by raw variance. In sequencing data, variance scales with mean expression. If a gene is highly expressed across the board, its raw variance will naturally be high, even if it is not biologically interesting. The algorithm has to decouple the variance from the mean expression. It does this by dividing genes into bins based on their average expression levels. Then it calculates a normalized dispersion within each bin. This tells you how much a gene varies compared to only other genes that are expressed at similar baseline levels. Scanpy offers different statistical methods to do this math, called flavors. The traditional CROT flavor expects your data to be log normalized first. It calculates dispersion, bins the data, and standardizes the values. There's also a newer CROT v3 flavor, which explicitly requires raw, unlogged count data to properly model the variance. Alternatively, the Cell Ranger flavor uses a slightly different approach to calculate normalized dispersion based on the counts. The flavor you choose simply dictates the specific statistical distribution used to model that relationship between the mean and the variance. When you run this function, it does not delete the rest of your data. Instead, it adds a few new columns to your variable annotations array. The most important one is a Boolean column simply called highly variable, marking true for the top 2,000 genes and false for the rest. Future steps in your pipeline will automatically look for this flag and only use those selected genes for downstream analysis. Here's the key insight. Feature selection is not just a computational trick to make your code run faster. It is the deliberate process of stripping away biological white noise so the true cellular identities have room to emerge. Thanks for hanging out. Hope you picked up something new.